So once again, thank you for the invitation from the organizers to come and speak to you about my work on Stenotrophomonas maltophilia. I want to start off by saying I'm coming from a primarily undergraduate institution, so the work I'm going to show you that's being done in my lab has actually been done mostly by undergraduate students. So it's taken a while to kind of get things up and running and optimized. So today I'm going to talk to you about the effect of select chemicals on S. maltophilia and its biofilm. So as I mentioned in my earlier talk, it's gram-negative, opportunistic pathogen, high mortality rate up to about 70% in immunocompromised patients, multi-drug resistance, a lot of it intrinsically based. It is a water contaminant, forms biofilms on moist surfaces. So I'm interested in the biofilm. And in my lab, I've got two research aims. One is to understand the mechanisms used by S. maltophilia to persist and form the biofilm. So how does the biofilm form? What are the triggers? What are the genetics behind it? My other research aim is to go in a slightly different direction and look for new ways, new approaches to reduce the biofilms and infections by the S. maltophilia. Because if you can inhibit the growth, inhibit the biofilm, you can reduce the chance of a patient acquiring this infection. Okay, so leading into this slide, I should mention I spent a number of months with my undergraduate students and a couple of grad students looking at different types of chemicals. So we looked at things, biocides, detergents, and eventually I ended up looking at surfactants. One surfactant in particular caught my attention. Is called polysorbate 80. This is the same thing as tween 80 in the lab. It is a non-ionic surfactant with detergent properties. It's a common additive in pharmaceutical preparations, so medicines commonly have tween 80 added at low concentrations. One of the nice things about the tween 80 is it's well tolerated in humans at low concentrations. The concentration I'm going to show you today that we're using is actually in that acceptable range. The surfactant itself is known to increase bacterial membrane permeability. These studies that I'm referring to here are on Pseudomonas aeruginosa. So as far as I know, no one's actually shown this in Stenotrophomonas maltophilia. So this is a slide showing some of the early work that we did in my lab, which is to look at biofilm formed by S. maltophilia. This is just a straightforward clinical isolate. This has actually come from a bloodstream of a patient. And looking at the biofilm after 24-hour development on polyvinyl chloride surface when grown in the lab with tween 80 at different concentrations at 37 degrees. So again, this is using the classical crystal violet assay to quantitatively measure the biofilm that's being formed. So as you can see here, in LB alone, Luria Botani broth, you get a nice strong biofilm. And as you increase the concentrations of tween 80, you do see a decrease in the amount of biofilm being made. You get down to around 0.05%, this starts to be significant. So I've started to use the tween 80 at this 0.05%. And I'm showing in different strains, different isolates, the same trend. If you give tween, it will inhibit the biofilm formation. So I ran across a paper by Sater and Jones in 2012 where they used surfactant polysorbate 80 to improve the MIC susceptibility testing results for polymyxins B and E. They show in this paper experimental work using a number of different strains of Pseudomonas aeruginosa, and they show that giving the polysorbate along with the Muller-Hinton broth in the typical micro-dilution plate assay, they show that it actually will improve the MIC susceptibility, having that tween present. So I was starting to connect the dots here and thought, okay, this is in Pseudomonas aeruginosa, does the same thing hold up in Stenotrophomonas? Because remember, they used to be related, they used to be in the same genus. So my first hypothesis was to see if the addition of polysorbate 80 at greater than 0.002%. The reason I use this is 0.002% is what the Sater paper published. So I'm using it at a concentration slightly higher than this and asking the question, will it lower the MIC for polymyxin B against S. maltophilia. Because S. maltophilia, remember, is well known for its low permeability of the membrane. It provides resistance to polymyxin B. So this is what we found. So this is work done by my undergraduates and myself. So polymyxin B, MIC results, using the typical assay as described by the Clinical Lab Standards Institute. We used Pseudomonas aeruginosa, ATCC strain, as a control for this experiment. These are 3 Stenotrophomonas strains. The first one is the reference strain. This has come from a CF chemotherapy patient. You can see MHB only, this is molar Hindenburg only, get an MIC up around 16 microgram per mil. If you add 0.05% tween 80 to that, it drops to about 8 microgram per mil. So it suggests that yes, it does increase the sensitivity of the organism to the drug. These 2 strains are ones that are clinical isolates. I chose them deliberately. This one is a respiratory tract isolate, and this one is a bloodstream isolate. I chose these because these represent the common infections of S. maltophilia. So again, you can see we do see a decrease as you increase the between 80% that's present. It is a subtle decrease, so it's not particularly significant, but you do see a slight decrease in the MIC. Okay, so my next experiment, my next hypothesis was the addition of this sorbate 80 to polymyxin B will inhibit the growth of the biofilm more than if you just try to treat S. maltophilia with polymyxin B alone. So I'm going to show you several graphs in a minute. We'll walk through the methods first. So we're doing the classical overnight growth culture. We're then taking it, standardizing it to OD of 1. Taking subcultures, growing them up for 2 hours with some shaking. This is important for aerobic agitation of the culture. 2 hours to get them into exponential growth, get them started off growing pretty well. Inoculate the polyvinyl chloride microtiter plate, wells with cells, with or without the treatments. Incubate the biofilm stationary 24 hours. Then remove the planktonic cells, measure the planktonic growth. Wash the biofilm twice. This is just washing it with sterile distilled water. And then stain, wash, measure using the crystal violet assay. We did 5 replicates per strain, 2 independent experiments, and we used ANOVA with multiple comparisons to look at the stats. So you're going to see several slides. They look very similar. You're going to see planktonic growth in the presence of the treatments. You're going to see from this side to that side. Typically it'll be LB only, so this will be the control that I'm measuring things, comparing different treatments to. This is the quantitative measure on the y-axis. Running on the x are the different treatments shown here. So for the K279 reference strain, LB only, you get a nice growth of the culture. If you add polymyxin B at 8 microgram per mil, I should mention the concentration of polymyxin B that I'm choosing is based on the MIC results that I showed you earlier. So I'm looking for differences. So 8 microgram per mil polymyxin B alone, we get some growth that's inhibited. We get a fair amount of growth still. If you add just tween by itself, at 0.05%, you see even more growth inhibition. If you combine these 2 things together, the polymyxin B and the tween 80 at that 0.05%, you definitely see a drop. You see a significant difference. And I should mention, for those of you not familiar with the stats, these letters, these small letters here, if you see different letters, it means these are significantly different from each other. Okay, so there's a definite trend here. Planktonic growth. Now let's look at what happens with the biofilm. So in the biofilm, we see the same trend. So the conclusion really is that addition of the polymyxin B here is increasing, addition of the sorbate 80 is actually increasing the efficacy of the polymyxin B. You get very, very little biofilm. This is about less than 2% of the biofilm remaining after these treatments. Okay, so moving on to the respiratory clinical isolate. So this has come from a patient. Again, we're using the same sort of treatments, only this time I'm including the treatment of just using tween 80 at 0.002%, because I wanted to mimic what that paper was telling me about increasing the MIC susceptibility. I wanted to see if the same trend would hold true. Would there be any difference between using tween 80 at 0.05% versus 0.002%? So again, you can see LB only, you get a nice growth. If you add a little bit of tween, not too much, 0.002%, you do see some inhibition, but essentially there's no difference between these. Okay, so that level of concentration of tween 80, not really making much impact on the growth. However, if you drop down to 0.05%, you definitely see a decrease in the growth. Polymyxin B alone, 5 microgram per mil, you do see some inhibition, but it's not as significant as adding just tween 80 at 0.05%. You combine the polymyxin B with the tween 80, you do see even further drop in the growth. But I can't see any significant difference between using the tween 80 at the low concentration or the higher concentration. But you can definitely say the addition of polysorbate 80 increases the efficacy of polymyxin B. Okay, so biofilm, similar results, except for one difference here, which is this. LB plus tween 80 by itself at 0.05% actually dropped the biofilm quite significantly. And it's interesting that adding the tween and the polymyxin together, a low concentration of tween and a higher concentration of tween, all three of these conditions drop the biofilm significantly compared to the other treatments, and yet I can't see a difference between these. So it suggests that the tween 80 itself can have an effect without adding the polymyxin B, but if you combine the two together, it's not going to hurt things. If anything, it'll help them slightly. So decrease the biofilm. So finally, this is the bloodstream isolate of Stenotrophomonas. Again, LB only. Growth we're measuring. 0.002%, so again, no significant difference here. Add even more concentration of the tween 80, you do see a significant decrease. Polymyxin B alone at 6 microgram per mil, you drop that growth even further. And then again, when you combine these two reagents, the polymyxin B, the polysorbate 80, you do see even further drops in the growth of this organism. So it's suggesting to me that, yes, the polysorbate 80 is able to increase the membrane permeability of Stenotrophomonas, thereby increasing the efficacy of the polymyxin B. So again, this looking at the biofilm of the bloodstream isolate, again, you can see the same general trend. And again, we can say addition of polysorbate 80 increases the efficacy of polymyxin B. And here we can't see any difference between the low concentration and the high concentration of the tween 80 itself. Okay, so this is interesting. This is all treating the biofilm as it's developing. Now let's look at the other scenario where the biofilm's already formed. Can we then treat that preformed biofilm and essentially remove it or disperse it? I'm going to show you data from one isolate to answer this question. So the hypothesis is the addition of the polysorbate 80 to polymyxin B will enhance the removal of preformed S. maltophilia biofilms. Again, the methods are very similar, except this time we're letting the biofilm form just in regular LB broth. Then we're washing the biofilm a couple of times. This is washed not in sterile distilled water, but in sterile phosphate buffered saline. We then treat the biofilm for 24 hours with some slow agitation. This is, it's roughly 125 RPMs, so it's a slow kind of rotating of the plate. We wash the biofilm twice after this 24-hour treatment. We wash this with sterile distilled water, then stain and measure the biofilm as I explained before. So again, we're using ANOVA with multiple comparisons to look at the data. So this is the strain. This is the respiratory tract strain. This is not really showing any difference because all I'm measuring here is the growth. So this is just growth in the wells just with LB. Just have the cells grown fairly consistently, have they formed a fairly consistent biofilm. This is prior to these treatments that are about to happen. So this should look non-significant, and we don't see any difference. Okay, so we've got a nice standard biofilm across the plate. Now after treatment, things change a little bit. So here's PBS only with the biofilm. That's our control. Phosphate buffered saline with low concentration and high concentrations of twine. Here they are. Not looking much different from PBS by itself. Then you start to look at what happens when you add in the polymyxin B. So polymyxin B by itself, yes, you do see some removal of the biofilm. Significantly different from these bars up here. And when you add in the twine at high concentration and low concentration, you do see removal of biofilm compared to these treatments right here. What is interesting, though, is the polymyxin B by itself is effective. So adding the twine helps, but it's not significantly different. I would ask you to keep in mind that this is a biofilm that's fairly sturdy, but I'm washing it several times. So I'm always conscious of shear forces, washing with the micropipette tips. So I'd like to repeat this data a few more times, but I think this is real. So again, the polymyxin B is effective alone. When you add in the polysorbate, it does help a little bit, but no real significant difference here. Okay, so in summary, polysorbate 80 lowers the MIC for polymyxin B against S. maltophilia, inhibits growth and biofilm formation of S. maltophilia, enables polymyxin B to be used more effectively to inhibit the growth in the biofilm. In combination with polymyxin B can help reduce preformed biofilms of S. maltophilia, and in combination with polymyxin B can be considered for use against infections. So in closing, I'd like to acknowledge my collaborators, particularly Dr. Tim Sparks, who helped me with the statistical analysis of the data. And these undergraduates here are people working in my lab currently. They were heavily involved in this experimental work, so they're responsible for the data that I'm showing you. Again, funding from NIH and DePaul University, well appreciated, and former graduate students in the lab who've worked with Stenotrophomonas. So thank you for your attention. I'll take questions.